Okay, so today I'm going to discuss a recent paper out of Cell Systems titled Indigenous Gut Microbes Modulate Neural Cell State and Neurodegenerative Disease Susceptibility. Okay, so I'm going to begin with some background information pertinent to this paper. Differential gene expression, a topic heavily explored in this paper. G protein signaling, likewise a topic explored. And temporality. I will begin with the background information. We have the gut-brain axis. We often hear people online discussing the importance of nourishing your gut microbiome. Okay, so yes, in our intestines we have bacteria and archaea, but bacteria are going to be of interest in this paper. So we have our intestinal cells. Okay, then we have specific bacteria. We have a wide variety of bacteria inhabiting our intestines. These bacteria, they have a manifold of effects. They could be beneficial or they could be harmful, but we are going to discuss the harmful outcomes. So we have these bacteria lining our intestines, and they produce products, metabolites from these substrate molecules, say stuff that we are unable to digest, and we are just going to assume that these metabolites are indeed absorbed, brought into circulation, eventually reaching the blood-brain barrier, crossing that, and entering cerebral tissue, because that is of interest in this paper. Alternatively, we can assume that the vagus nerve, which originates in our brainstem, sends down these receptors, these afferent fibers, down into our gut, and these fibers actually respond to stimulation from what is going on in our gut. So cells in our brain respond to these signals to perform a function. All right. Okay, and real fast, I'm going to discuss germ-free. When we are germ-free, we lack microorganisms, and this was really explored by Keen et al. In animal model studies, where mice that lacked these microorganisms demonstrate cell immaturity. Cell immaturity arises when cells do not receive specific signals from, say, gut microbes. These gut microbes elicit effects that cause these cells to respond. All right, and the response by these cells causes them to follow specific pathways, and this allows them to perform specific functions which they are expected to perform in ideal conditions. So in the absence of these signals, they don't perform specific functions because they lack the specific stimuli that causes them to perform that function. And the reason I continue to mention cerebral tissue is because this paper really emphasized the cerebral tissue and the hippocampal nuclei. So I'm going to be discussing brain resident immune cells. To begin, differential gene expression, as I kind of hinted at earlier, occurs when a cell receives a signal, and this signal could either be intrinsic or extrinsic, but specifically of interest is going to be the extrinsic signals, of course, which arise from the gut microbiota, so the microorganisms in the gut. And when they receive this signal essentially acts as a ligand. Okay, so it is a stimuli that binds the cell, binds a receptor. This is going to end up activating, say, a given protein, an effector, and this is going to elicit effects that cause the cell to engender specific products that allow it to perform a specific function. Okay, so once again, this stimuli, this metabolite, is going to bind a cell, say in your cerebral tissue, end up activating a specific effector. That effector is going to bind somewhere on the DNA, and the DNA is going to be transcribed. That is going to give rise to mRNA. That mRNA is going to give rise to a protein. That protein is then going to perform a specific function specific to that cell. All right, we don't observe specific proteins across every cell type because we want our cells to be specialized. That said, these metabolites likely only bind to receptors on this specific cell type. All right, so these metabolites that induce these specific effects will likely not affect, say, cell types in your eye. And this paper specifically explored, say, monocolonization. Okay, so colonization of our gut by specific strains of bacteria. And these bacteria were selected based on the natural prevalence in the human gut. However, it should be noted that these bacteria all elicit different effects, and these effects were actually interestingly realized to be inversely related or independent of each other. For instance, we could talk about E. coli and its tendency to induce adaptive immunity via its stimulation of T cell differentiation. Say we took a different bacterium this was observed to counteract these effects. Not counteract these effects necessarily, but to just produce the inverse effect. So that was really cool to observe. Okay, so to stay on this topic, we're going to discuss E. coli and its relationship to neurodegenerative disease, specifically that it influences transcriptional responses that are conducive to the development of such ailments. So, adaptive immunity, related to what I was just mentioning about the T cells, we see an increase in CD8 plus cytotoxic T cells in the brain and a decrease in the peripheral CD4 plus helper T cells. Likewise, we observe an increase in the phagocytic activity and an increase in the disease-associated microglia, and this is temporal. What this means is that this is not an overnight occurrence, rather it occurs over time. So, presumably, we observe an increase in phagocytic activity and an increase in the cytotoxic T cells because we want a mechanism, a natural mechanism, to combat the formation of these amyloid beta plaques. Alright, we want to phagocytose, we want to get rid of these harmful plaques. Okay, and we know from previous studies that Alzheimer's disease is related to the concentration of E. coli in our gut. Okay, and real quickly to relate this to animal model associations, we observe in these animal models, okay, so the 5X FAD mice, that they actually exhibit an increase in CD8 plus T cells, and that was mentioned because there's often an association of CD8 plus T cells with Alzheimer's disease. And number 4 of interest is going to be the changes in cognitive function. And this is going to arise from animal models, which was explored in this study. And we observed that there is an increase in primary latency, which is essentially a measure of cognitive function, okay, and like in a maze assessment that they do for these mice. Additionally, we observed that there is an increase in alpha beta plaque density, which is inversely related with memory assessment. So yes, this increase in plaque density was indeed observed in these E. coli monocolonized mice. However, there was no increase in the number of plaques, just the actual density of the plaques, which is interesting. And the one thing that really bothers me is that we don't know the mechanism by which this actually occurs. We just know that the relationship between E. coli monocolonization, so the presence of E. coli, is associated with these pathologies. Okay, so to briefly summarize these findings, we observed that E. coli elicits transcriptional responses, which is associated with increased phagocytic activity and increased adaptive immunity. And we know that the concentration of E. coli is associated with Alzheimer's disease pathology, associated with decreased memory functionality, increased concentration of these plaques, however not an increase in the number of these plaques. And the observed changes in adaptive immunity likely result in an increased phagocytic activity toward these amyloid beta plaques. However, the mechanisms and the actual reasons for these increases is yet to be explored. To discuss another facet that was also explored related to E. coli monocolonization concerns G protein signaling. It was observed that there is a decrease in Rho-dependent GTPase signaling. So all the effects that could result from, say, a ligand binding a cell, activating this G protein, which gives rise to countless effects, is simply not observed because this signaling pathway is not occurring. Alright, so what that means is that these effects that E. coli likely elicits in a given cell are numerous and uncountable. And the reason that I mentioned that it is likely improbable to actually measure all the effects of the decreased Rho signaling is because it is essentially analogous to, say, a given person sharing a rumor to, say, a friend, and then that friend sharing it to another person, then that person and those other people sharing it to more people. So it's just like this cascade that is endless, and everything is just linked together because you likely know the person that someone else shared that to. So it's just trying to trace that back to the origin is just pretty much implausible. And because this process essentially goes on perpetually, it's kind of hard to trace it back to the origin. So yes, there's that. To move on and discuss temporality related to E. coli monocolonization, we know that differential gene expression takes time. Pathway changes were observed in this study. There was a decrease in mRNA processing at 2 weeks, and then 2 weeks later, there was an aerobic respiration decrease as well as oxidative phosphorylation decrease. Okay, so forgive my speculation here, but I'm going to assume that these mRNA that were supposed to be being processed were actually precursors to the mRNA that were going to be implicated in these processes, alright? Akin to how we would have, say, in the baking of a cake, alright, a given person wants to bake a cake. we want to perform aerobic respiration, we want to utilize oxidative phosphorylation. All right. However, if we say lack eggs for that cake, all right, so if we lack mRNA, which were precursors, weren't being produced, we no longer have the eggs to make this cake, we no longer have the eggs, we no longer have the mRNA to perform these processes. So it is a temporal process. It occurs over time. We lose the intermediates to perform these higher order functions, because we recall that cells function to maintain homeostasis, so this is a homeostatic process. Alright, and I'm going to finally begin discussing sex specificity, which was observed. So we observed that pre and postnatal discrepancies are a thing when it comes to differential gene expression, and this was explored earlier by Theon et al. In 2018. So what I mean by that is, say males in utero have different patterns of gene expression than females. Okay, so say that a male in utero is highly sensitive to the concentration of E. coli, whereas postnatally females are more sensitive, so they have different effects at different time periods, which really limits the findings and may ultimately complicate them, which is unfortunate. And we also have sensome genes, which likewise vary between sexes, which further complicate things, in which these sensome genes are related to stimuli from the environment, so they are likely intimately associated with this process, because we know that external stimuli is an extrinsic factor, so there is that. And these differences are at the cellular level, which is consistent with what we've been discussing throughout this paper. Okay, and real quickly to discuss my concerns with this paper, and just more generally this field of research, is that the mechanisms have yet to be explored. This paper, however, did roll out one of them, which concerned inflammation. Inflammation was assumed to be related to this process. However, there was no observed change in colon length, because we know that when tissue becomes inflamed, it typically, say, swells, so it changes in size. Likewise, to rule out the potential relationship between E. coli concentration and inflammation, there was no significant change in the levels of cytokines, chemokines, and other molecules associated with inflammatory pathways. So yes, these inflammatory markers are unlikely to be responsible for eliciting these transcriptional effects realized. So yes, researchers are still in the weeds to look for a mechanism that is resulting in these changes in transcriptional responses, which is why I've operated my thinking based on metabolites that are produced and travel and traverse the BBB and elicit effects. Okay, and another concern of mine is the actual harmful or benefits of these gut microbiota. E. coli were explored, and we know that each of them produce their own individual effects, and these effects are countless and often unquantifiable. But because this study really pigeonholed on just E. coli, all of the other bacteria, all of the other common bacteria have really yet to be explored in terms of their actual benefits. But this is only likely to manifest after researchers explore all of the different kinds of bacteria that are likely to reside in our intestines. But it is really cool that we can actually potentially in the future alter this microbiome to elicit effects that may be beneficial to the host. But yeah, that is all I wanted to discuss today, so keep staying alive.